Hello, and welcome to this Anatomy Physiology Bio 202 video lecture on development and inheritance. I'm Mr. Kennedy, and I'll be your guide as we explore these topics together. We'll begin by looking at the embryonic period of human development. The embryonic period extends from fertilization through the 8th week of development. Fertilization occurs when sperm and secondary oocytes merge. The sperm swim from the vagina to the cervix using their tails. They then pass through the uterus and uterine tubes, mainly due to the contraction of the walls of these structures. To fertilize an egg, sperm must penetrate the corona radiata and the zona pellucidea. This is an illustration of that event. The enzymes of the sperm's ectrodrome, along with tail movement, allow the sperm to penetrate the corona radiata. Glycoprotein ZP3 in the zona pellucidea is a receptor for sperm. The membrane proteins in the sperm head bind to ZP3, and acrosomal enzymes are released to digest a path in the zona pellucidea. This is that event occurring under a scanning electron microscope. The haploid nucleus in the head of the sperm becomes the male pronucleus. The haploid nucleus of the fertilized ovum becomes the female pronucleus. When the two merge, it's referred to as syngamy. The diploid zygote is formed. After fertilization, at about 24 hours, the zygote begins mitotic division called cleavage. The first division takes about 6 hours. Successive divisions take much less time. By the second day after fertilization, a second cleavage is completed, yielding 4 cells, illustrated here. By the end of the third day, there are 16 cells. Each division yields smaller and smaller cells. By the fourth day, the cluster of cells resembles a mulberry and is called a morula. It's still surrounded by the zona pellucidea and still the size of the zygote. On day 4 or 5, the morula enters the uterine cavity and is nourished by uterine milk, a glycogen-rich secretion from endometrial glands, in addition to stored nutrients from the cytoplasm. At the 32-cell stage, the fluid now inside the morula rearranges the blastomeres into a large, fluid-filled blastocyst cavity. The mass is now called a blastocyst, still the same size as the original zygote. This is an illustration of the external view of a blastocyst on day 5. As the blastocyst formed, 2 different cell populations arose. The embryoblast, which is the inner cell mass and will develop into the embryo, and the trophoblast, or the outer cell mass, which will develop into the outer chorionic sac surrounding the fetus and the fetal portion of the placenta. This is the sectioned view of the blastocyst at day 5. The blastocyst remains free in the uterine cavity for about 2 days and then implants by attaching to the endometrium at around 6 days after fertilization. Implantation usually occurs in either the posterior portion of the fundus or the body of the uterus. The inner cell mass orients towards the endometrium. After implantation, the endometrium is called the decidua. It separates from the endometrium after the fetus is delivered. The decidua has different regions named based on their positions relative to the site of the implanted blastocyst. This graphic illustrates the points or events that occur leading to implantation. Fertilization occurs, within the uterine tube 12 to 24 hours after ovulation. Cleavage occurs about 30 hours after fertilization. By day 3 or 4, we have a morula. By day 6, the morula has advanced through its blastocyst stage and implanted. About 8 days after implantation, the trophoblast develops into a syncytotrophoblast and cytotrophoblast. At around 8 days, the embryoblast also develops into 2 layers, the hypoblast and epiblast. Cells of these structures form a flat disc called the bilaminar embryonic disc. The amniotic cavity forms from the epiblast. Those structures are seen illustrated here. The amnion forms from the roof of the amniotic cavity. Eventually, it surrounds the entire embryo and fills with amniotic fluid. Also, on the 8th day, the exosomic membrane forms that, together with the hypoblast, forms the yolk sac. On the 9th day, small spaces called lacuna form. By the 12th day, they fuse to form lacunar networks, as seen here. About the 12th day after fertilization, the extraembryonic mesoderm develops. The cells form a connective tissue layer around the amnion and yolk sac. Large cavities develop that fuse and form the extraembryonic coelom. The extraembryonic mesoderm, together with the trophoblast, forms the chorion, which surrounds the embryo and later the fetus. The chorion blocks antibody production by the mother, promotes the production of T lymphocytes to suppress the immune response in the uterus, and produces human chorionic gonadotropin, or HCG. The first major event of the 3rd week of development is gastrulation. The 2-layered embryonic disc transforms into a trilaminar or 3-layered embryonic disc made up of ecto, meso, and endoderm. Gastrulation is associated with the rearrangement and migration of cells from the epiblast. The first step in gastrulation is the formation of the primitive streak. That's illustrated here. The primitive streak establishes the head and tail ends of the embryo. Next, cells of the epiblast move inward below the primitive streak and undergo invagination. Following this, the 3 germ layers form, as illustrated here. This table will provide you with a summary of the structures produced by the 3 primary germ layers, endo, meso, and ectoderm. About 16 days after fertilization, the notochord processes form. By days 22 to 24, the process becomes the solid cylinder called the notochord. The notochord is important for induction, the process whereby the inducing tissue stimulates development of a responding tissue to develop into a specific structure. The notochord induces the development of vertebral bodies and the nucleus propulsus of vertebral discs. That is illustrated here. Also, during the 3rd week of development, the following structures form: oropharyngeal membrane, cloacal membrane, and the allantois. The notochord also induces development of the neural plate, illustrated here. The plate develops the neural fold as the lateral edges become more elevated. The depression, or depressed mid-region of the fold, is the neural groove. As the neural folds approach each other and fuse, the neural tube is formed. The process for the formation of all these structures is referred to as neurulation. As the neural tube forms, some of the ectodermal cells from the tube migrate to form several layers of cells called the neural crests. That's illustrated here. At about 4 weeks after fertilization, the head end of the neural tube develops into 3 enlarged areas called primary brain vesicles, as seen in the diagram below. The vesicles are called the proencephalon, or forebrain, mesencephalon, midbrain, and rhombencephalon, or hindbrain. By about the 17th day after fertilization, paired, cube-shaped structures called somites form. By the end of the 5th week, 42 to 44 pairs are present. Each somite differentiates into a myotome, a dermatome, and a sclerotome, as illustrated in this diagram. At the beginning of the 3rd week, the formation of blood vessels begins with the development of blood islands. This is an illustration of blood island development. On days 18 and 19, the heart begins to develop in the head end of the embryo. It begins as a region of mesodermal cells called cardiogenic area. A pair of endocardial tubes forms. The tubes fuse to form a primitive heart. This illustrates the formation of the heart. Embryonic tissue invades the uterine wall and erodes uterine blood vessels. Blood fills spaces called lacunae, and by the end of the 2nd week, chorionic villi develop, as illustrated in this picture. By the end of the 3rd week, the blood vessels develop into chorionic villi. They connect to the embryonic heart. The vessels connecting to the heart do so by way of the umbilical arteries and umbilical vein through the body stalk, which eventually becomes the umbilical cord, as illustrated here. Placentation is the process of forming the placenta. This structure is the site of exchange of nutrients and waste between the mother and fetus. The placenta produces hormones used to sustain the pregnancy. By the beginning of the 12th week, the placenta has 2 parts, the fetal portion, or chorionic villi, and the maternal portion, the decidua basalis of the endometrium. Those structures are illustrated in this diagram. When fully developed, the placenta is shaped like a pancake, and it is able to protect the fetus from microorganisms as well as its other functions. All major organs develop between the 4th through 8th weeks in a time period known as organogenesis. Embryonic folding occurs during the 4th week. This involves the flat embryo folding into a 3-dimensional cylinder. The head fold and a tail fold develop at about day 22. Lateral folds form as they move toward the midline. They incorporate the yolk sac into the embryo as the primitive gut. On the outside of the embryo is a cavity in the tail region called the proctoderm. Separating the cloaca from the proctoderm is the cloacal membrane. 5 pairs of pharyngeal arches, or branchial arches, also develop on each side of the future head and neck regions during the 4th week. Each arch is separated by a pharyngeal cleft. Pharyngeal pouches meet the pharyngeal clefts, as illustrated here. By the middle of the 4th week, upper limb buds begin to develop. By the end of the 4th week, lower limb buds and the heart prominence form. At the end of the 4th week, the embryo has a tail. During the 5th week, the brain and head develop rapidly, and the limbs develop further. By the 7th week, the regions of the limbs become distinct and digits appear. By the end of the 8th week, eyelids come together. The tail disappears. External genitals begin to differentiate, and digits are distinct and no longer webbed. The fetal period continued. The fetal period begins at the 9th week after fertilization. Tissues and organs that develop during the embryonic period grow and differentiate. Very few new structures appear during this period. This is a graphic illustrating the process of development that occurs during the fetal period. And this table will provide you with a summary of changes during the embryonic and fetal development. The time period, approximate size and weight, and representative changes are all listed. Next, we'll examine a few threats to the fetus, such as teratogens. Teratogens are any agent or influence that is able to cause developmental defects in an embryo or a fetus. Any number of chemicals and drugs may be considered teratogens. Alcohol is the most common, causing fetal alcohol syndrome. Others include viruses, industrial chemicals, some hormones, antibiotics, cocaine, and many others. Cigarette smoking during pregnancy has also been implicated as a cause of low infant birth weight, cardiac abnormalities, anencephaly, and higher infant and fetal mortality rates. Ionizing radiation in many forms is also teratogenic. Exposure of the mother to X-rays or radioactive isotopes during pregnancy may cause microcephaly, a small head, mental retardation, and skeletal deformities. During pregnancy, several medical tests are used to detect fetal abnormalities, genetic disorders, and overall well-being. Fetal ultrasonography is used to determine a more accurate fetal age when the date of conception is in doubt. It's also used to confirm pregnancy, determine fetal position, identify multiple pregnancies, along with other uses. Amniocentesis involves the removing of some amniotic fluid surrounding the developing fetus and analyzing it and fetal cells for genetic abnormalities. It's usually performed between weeks 14 and 18. The needle used to collect the fluid is guided by ultrasound to avoid damage to the fetus or the umbilical cord. Chorionic villus sampling may be performed as early as 8 weeks of gestation. It is also done under ultrasound guidance, but the usual procedure is to insert a catheter through the vagina and cervix to collect a tissue sample from the chorionic villi. The goal is to identify the same genetic defects as seen with amniocentesis. The procedure may be done through the abdominal wall, as with amniocentesis. Non-invasive prenatal tests may also be performed, but they are currently not as informative as amniocentesis and chorionic villus sampling. The maternal alpha fetoprotein, or AFP test, requires a blood sample from the mother. It's used to detect AFP, a protein produced by the fetus at its highest levels between weeks 12 and 15. After the 16th week of pregnancy, when levels go to zero, high levels of this point indicate a neural tube defect. During the first 3 to 4 months of pregnancy, the corpus luteum secretes progesterone and estrogens in low levels. From the 3rd month to the end of pregnancy, the placenta produces high levels of these hormones. The chorion secretes HCG to stimulate the corpus luteum to produce estrogens and progesterones and inhibit menstruation until placentation takes over. This is a graphic that illustrates some changes in the maternal body during pregnancy. And this graphic illustrates hormone changes in the mother during pregnancy. HCG levels will peak at about the 9th week of pregnancy. The chorion will secrete estrogens after the first 3 or 4 weeks of pregnancy and progesterone by the 6th. Relaxin is secreted by the corpus luteum and later by the placenta. It increases the flexibility of the pubic symphysis and the ligaments of the sacroiliac and sacrococcygeal joints. It also helps dilate the cervix during labor. Human chorionic somatomammotropin, also known as human placental lactogen, probably helps prepare the mammary glands for lactation and helps the maternal growth and regulates metabolism in mother and fetus. The hormone recently discovered to be secreted by the placenta, corticotropin-releasing hormone, it is secreted in non-pregnant people by the hypothalamus. It is involved in the timing of birth. CRH is also needed to increase secretion of cortisol, which is needed for the maturation of fetal lungs and the production of surfactant. The uterus continues to expand throughout the pregnancy, moving upward into the abdominal cavity until it almost fills it. The organs are pushed out of the way, and pressure on the stomach may cause food to be displaced, causing heartburn. This is a graphic illustrating that event. Labor. Labor is the process that expels the fetus from the uterus through the vagina. Labor is initiated by the interaction of several hormones. Control of contractions occurs via a positive feedback cycle. True labor begins when uterine contractions occur at regular intervals. False labor is associated with irregular contractions and no-show, a discharge of blood with mucus. True labor is divided into 3 stages: dilation, expulsion, and placental stage. This is a graphic illustrating labor. Following delivery, it takes about 6 weeks for the maternal reproductive organs and physiology to return to the pre-pregnancy state. This period is the puerperium. The reduction in size of the uterus is involution. Adjustments of the infant at birth. During development, the baby is totally dependent on the mother for survival. At birth, the fully developed newborn body begins to function independently. At birth, the lungs are able to exchange oxygen and carbon dioxide thanks to surfactant that began to develop by the end of the 6th month. The respiratory rate at birth is 45 breaths per minute, dropping to the normal 12 breaths per minute within 2 weeks. After the baby's first breath, many changes must be made in the cardiovascular system. The foramen ovale closes to become the fossa ovalis. The ductus arteriosus closes to become the ligamentum arteriosum. The umbilical arteries fill with connective tissue. The umbilical vein becomes the ligamentum teres of the liver. Those changes are illustrated here. The physiology of lactation. Lactation is the production and ejection of milk from the mammary gland. Prolactin, which is secreted by the inferior pituitary gland, is the main hormone in stimulating milk production, while oxytocin causes the release of milk into the mammary duct via milk ejection reflex. Those processes are governed by a feedback mechanism illustrated here. Inheritance. Inheritance is the passage of hereditary traits from one generation to the next, and genetics is the study of inheritance. Humans have 23 pairs of chromosomes, 1 in each pair from the father and 1 from the mother. Genes for the same trait that are in the same location on each homologue are referred to as alleles. A mutation is a permanent heritable change in an allele. One genetic disorder caused by a mutation is phenylketonuria, or PKU. People with PKU cannot make the enzyme phenylalanine, which is needed to break down phenylalanine. A Punnett square is used to show the possible genes inherited from two parents. The genotype is the actual genetic makeup related to a trait. An allele that dominates or masks the presence of another allele is a dominant allele represented by an uppercase letter. The allele whose presence is completely masked is the recessive allele, represented by a lowercase letter. Phenotype is the physical expression of the genotype. This graphic illustrates the use of a Punnett square to predict the probable outcome of a genetic cross. And this table will provide you with a summary of dominant and recessive traits in humans. Most patterns of inheritance don't conform to simple dominant-recessive inheritance patterns. Incomplete dominance is a situation where neither member of the pair of alleles is dominant over the other. An example of incomplete dominance is the inheritance of sickle cell anemia. This graphic illustrates the inheritance pattern for sickle cell anemia. Multiple allele inheritance occurs when genes have more than two alternative forms. Inheritance of the ABO blood group is an example of this. Within this inheritance pattern, there's also codominance. In this case, two genes, type A and type B blood, are expressed equally. This graphic illustrates blood type heredity. Polygenic inheritance is seen when a trait is controlled by the combined effects of two or more genes. Complex inheritance is seen when a trait occurs due to the combined effects of many genes and environmental factors. Examples of complex traits include skin color, hair color, eye color, height, metabolic rate, and body build. This graphic illustrates complex hereditary patterns. The 46 human chromosomes, or 23 pairs, are identified by their size, shape, and staining pattern. An entire set of chromosomes arranged in decreasing size order and according to the position of the centromere is called a karyotype. This is an illustration of a karyotype. The 23rd pair of human chromosomes is our sex chromosome. The 22 preceding pairs of chromosomes are referred to as our autosomes. Males have an X and a Y chromosome at the 23rd position in their karyotype, while females have two X chromosomes. One X chromosome is automatically inactivated and becomes a bar body. Whether the sperm that will fertilize an egg is carrying an X or a Y chromosome will determine the gender of the zygote. An egg will only have one X chromosome under normal conditions. This graphic illustrates the inheritance patterns associated with the 23rd position in a human karyotype, or the inheritance of the sex chromosomes. Some non-sexual traits are inherited on the X chromosome. These are called sex-linked traits. An example would be red-green color blindness. This is an example of a Punnett square that illustrates the hereditary pattern of a sex-linked trait. And this concludes our coverage of development and inheritance.